Hello everyone, my name is Ronald Ngetich and I'm representing my co-authors Wenjuan Li and Professor Ling Li from the University of Electronic Science and Technology of China. The title of our poster is Inhibiting the Visuospatial Working Memory Performance using Continuous Data Bus Stimulation over the Dorsolateral Prefrontal Cortex. The dorsolateral prefrontal cortex plays an important role in visuospatial working memory. However, its hemispheric specialization in this cognitive process remains unclear. While TMS has been demonstrated to be an effective method of neuromodulation, a specific variant of TMS, the CTBS, has been shown to inhibit cortical activity of the targeted brain areas. Below is how the TMS machine works. Our experiment consisted of 6 subjects, 18 each for the left and the right DLPFC groups. For the right DLPFC group, we stimulated the right DLPFC whose MNI coordinates are as shown and used vertex as the control site. For the left DLPFC group, the participants received CTBS over the left DLPFC with vertex as the control site. We used a uniform intensity of 40% of the maximal stimulator output for all subjects. This is the pattern of the CTBS. Experimental design. Our experiment consisted of 2-back and 4-back visuospatial working memory tasks. In the 2-back task, the subjects were required to remember the square displayed in the 2 positions back and to assess whether the currently presented square is in a similar position, and if so, they were required to press the button 2. If not same, not to respond. The same case for the 4-back task. The subjects were required to judge whether the currently displayed square is at the same position as that of the 4th position back. If same, they were required to press key button 2. If not the same, not to respond. Results. Our D' index results for the left DLPFC group suggest that the performance was not significantly affected either in 2-back or in 4-back. Conversely, the CTBS over the right DLPFC significantly impaired performance in 2-back task. So this one in 2-back task relative to the vertex or the baseline. However, 4-back task performance was not significantly affected by the CTBS regardless of the stimulation condition. For the CTBS effect, to get the CTBS effect, we subtracted the baseline D' scores from the post-stimulation D' scores. For the left DLPFC group, we did not observe any significant CTBS effect, both in 2-back and in 4-back tasks. For the right DLPFC, there was a significant CTBS effect after the CTBS over the right DLPFC relative to the vertex in 2-back task. However, there was no significant stimulation effect observed in the 4-back tasks. In conclusion, our findings demonstrate the effectiveness of CTBS in modulating visuospatial working memory performance and highlight the crucial role of DLPFC, especially the right DLPFC, in this cognitive function. Taken together, our results may be of great importance in the study and treatment of neuropsychological conditions that are characterized by a deficit in visuospatial working memory. Thank you.